Hello student. So up to the last class or last episode we have discussed about the general introduction of gene interactions. Now today we will elaborate them. So let's take some examples of the gene interactions. Incomplete dominance. Example, flower color in Mirabilis jalapa, four o'clock plant popularly called. In this plant, red flower plant capital R is allowed to crossed with white flower plant small r. In F1 generation, all plants are of a pink flower. And when the genotype capital R small r, when they allow for the selfing, half of the gametes capital R, half of the gametes small r. Similarly, half of the gamete capital R, half of the gamete small r. And now the progeny capital R, capital R, capital R, small r, capital R, small r, and small r, small r. It means red flower, it means pink flowers, it means pink flowers, and it means white flowers. Means in F2 progeny, this is F2 progeny, this is F1 progeny, these were the parents. So in dihybrid cross, you are watching that, in incomplete dominance case, you are watching that the red flower plant when crossed with the white flower plant, instead of the red flower in the F1 generation, we are obtaining pink flower. While you know it was not the case of Mendelian genetics. In case of the Mendelian genetics, the F1 heterozygote should be identical to the one of the homozygote. Here F1 heterozygote is the intermediate of both of the homozygotes. That is the deviation of Mendelism. That is the deviation of dominance concept, and this is called incomplete dominance. Here there in F2 progeny, phenotypic ratio that we are obtaining is the red is to pink is to white, 1 is to 2 is to 1, while in Mendelian genetics phenotypic ratio was the 3 is to 1. Genotype is same, 1 is to 2 is to 1. Means in case of gene interactions, genotype not change, only phenotype get changed, external appearance get changed. Rather than the 3 is to 1, now we are obtaining 1 is to 2 is to 1, means we are deviating to the Mendelian genetics or Mendelian principle of inheritance. This is incomplete dominance. Why it is going on? Look at here. Incomplete dominance is a kind of monogenic quantitative character. Means here there the character is not governed by the presence of capital R or absence of capital R. Here character is governed by the protein product, amount of the protein product arise from the capital R. In case of capital R, there are two capital R present. So suppose the pigment which is required for the flower color arise in the X amount. If X amount of pigment arise from the capital R and capital R, that is why color of the flower would be red. That in capital R small r conditions, the number of R instead of the 2 is a 1. So the amount of pigment would be half, means x by 2 pigment arise. Because x by 2 pigment arise, it means as compared to the red color, the color would be comparatively less. So the color appear pink. And in the third case where there completely their R is missing, capital R is completely missing, that is why the amount of pigment is 0. If amount of the pigment is 0, so the color of the flower is also 0, means colorless, means white. Means here character is governing by the number of dominant gene. If 2 dominant alleles are present, red color. If 1 is present, pink color. If 0 dominant gene or allele is present, then the colorless condition, means character is controlled by the quantity of the product or number of the dominant alleles. That is the incomplete dominance. Similarly, here there only one case you studied, that is the flower color in Milapis jalapa. One more case is mentioned in our NCERT, that is also I am going to discuss with you. Second example that is mentioned in our NCERT about the incomplete dominance is the size of starch grains in pea plant. In pea plant, if capital B gene is present in homozygous condition, then the large sized grains of the starch arise. And if capital B small b condition is present, then intermediate size of the starch grains arise. And if small b is the condition, means none of the dominant allele is present, then the small sized starch grains will be obtained. It means here the character is not distinguished only in two forms, large and small, large, intermediate and small. Means here there also intermediate phenotype is arising. So this is also a case of incomplete dominance. I would like to elaborate one of the paragraph of the NCERT which is mentioned about this particular concept, starch branching enzyme gene. Suppose I am denoting it as a capital B. When go through the process of transcription and translation, gives rise to the starch branching enzyme. Starch branching enzyme, if present, then it will regulate to the two phenotypes. It will regulate to the shape of seed as well as size of starch grain. Means here there one gene regulating to the more than one phenotype. Whenever one gene regulates to the more than one phenotype, this condition is known as a pleiotropy. Means this starch branching enzyme gene is not only the case of, or this is not only the case of incomplete dominance, this is the case of pleiotropy also, that we will study into the coming hours. Anyway, so shape of the seed if concerned, then with respect to the gene B, there are three combinations are possible: capital B combination and capital B small b combination. Both of the combination will give rise to the round seeds. But small b condition will gives rise to the wrinkled seeds. In contrast, about the size of starch grain, capital B gives rise to the large sized grains. Capital B small b condition, intermediate grains. And small b means small sized grains. Means you just think about it that this capital B gene is regulating to the more than one phenotype, and for this phenotype, if this phenotype is in our consideration, then this gene is showing behavior of the dominance. Because capital B as well as capital B small b both are showing what? Both are showing dominant phenotype. Means with respect to the shape of the seed, gene B is dominant over the small b. But if we consider this phenotype, size of the starch grain, then what you are watching, that capital B gives rise to the large size starch grain, and capital B small b, unlike shape of the seed, is here giving rise to the intermediate size of the grains. It means with this respect, gene B is showing incomplete dominance, while with respect of this phenotype, it is showing dominance. It means you can say likewise that dominance is not an autonomous property of the gene. Dominance is not an autonomous property of the gene, because if dominance is an autonomous property of the gene, then whatever the phenotype it regulates, the dominance should also be reflect. But here we are watching that in this case there is a dominance, but in this case there is no dominance, while regulation of both of the phenotype is by the same gene. It means dominance is not an autonomous property of the gene. Dominance depends on the gene, dominance depends on the product of the gene, but dominance depends on the development of the phenotype from the gene. But beside this, it also depends that what phenotype you choose for the examination. If for the examination of the dominance you are choosing this phenotype, then there is a dominance. But for the examination, if you are choosing this phenotype, then there is an incomplete dominance. So dominance depends on the gene, depends on the product of the gene, depends on the production of the phenotype from the gene. But beside this, it also depends on the selection of the phenotype that you are using, that you are choosing for the examination. That is one of the crucial paragraph given in our NCERT. I hope now you would be in a more comfortable region. Some other examples of the incomplete dominance also we can visualize in our nature, like feather color in Andalusian fowls is also one of the example of incomplete dominance. Similarly, some other cases of the incomplete dominance is also existing, but these two typical examples, pea plants starch grain size and the Mirabilis jalapa, are the very crucial examples that should be in your notice. Just remember, it is a kind of a monogenic quantitative character. Mendelian genetics was monogenic qualitative. Here it is a monogenic quantitative. Okay, yes, phenotypic ratio 1:2:1, while in Mendelian genetics it was the 3:1. Genotype is same for both. Now second type of a gene interaction, intragenic, that was the codominance. Codominance. In incomplete dominance, what I said, that the phenotype is intermediate of phenotype of the heterozygote is intermediate of the phenotype of both of the homozygotes. Now here in codominance, what happened? Phenotype of the F1 would be determined by both of the genes coming from both of the parents. How? Look at here. I am taking an example of the hair colors in cattle. Red-haired cattle R1, R1. If allowed to cross with white hair cattle, R2, R2. Intentionally, rather than the pattern of the representation of alleles as capital letter and small letter, I am using a different pattern. Reason is what? Because in case of dominance, no one is dominant and no one is recessive. If no one is dominant, no one is recessive, both of the allelic variant of this gene are equally expressed, then how I can represent them as one with the capital letter and one with the small letter? That is not the logical one. That is why I am using a different pattern of the presentation of allelic forms. One of the allelic variant I use with R, I represent with the R1, and second variant of it I am using as a R2. So this is homozygous R1 R1 like capital T, and this is homozygous R2 R2 like small t. Anyway, now the F1 progeny that will obtain, that is known as a roan cattles. Roan cattles means what? On the body there is somewhere there is a patch of the red hairs, somewhere patches of the white hair, red hairs patches, white hair patches. That kind of a mosaic body is known as a roan character. Means all are genotypically R1 R2. Now they are allowed for the sib mating. In case of the animals, we will use the word sib mating, means crossing between the siblings, sib mating. While in case of the plant, what the term we were using? Selfing. Yes. So here there two types of gametes, half of the gametes R1, half of the gametes R2. Similarly, half of the gametes R1 and half of the gametes R2. This is R1 R1, this is R1 R2, this is R1 R2, and this is R2 R2. Means red haired cattles we obtain, roan cattles we obtain, and white haired cattles also we obtain in the ratio of 1:2:1. Genotype is same. Means here there also we are obtaining to the phenotype 1:2:1 like in case of incomplete dominance. But one thing should be always in your mind, that here both of the allelic variant R1 and R2 are equally expressing within the same organism simultaneously. So the word arises codominance. While in case of incomplete dominance, only R, capital R was expressing, but the expression was complete if both of the alleles are capital R, and it was incomplete when one is the capital R, second is the small r. So there was the incomplete expression of the dominant variant. There is an equal expression of both of the variant within the same organism. So the word is a codominance and the word was the incomplete dominance. Phenotypic ratio for both would be same, 1:2:1, and there was also 1:2:1. Don't judge any interaction on the basis of just their phenotype. That here is also 1:2:1 sir, there was also 1:2:1, it means both should be the same interaction. No, you must remember 3 plus 3 is all 6 and 7 minus 1 is also 6. Resultant of both is a 6, but one is an addition and second is what. Anyway, on the basis of the incomplete about the codominance, some more examples are here there in our nature. You heard about the blood group AB. In ourselves, there is a ABO blood group system in which 4 types of phenotype you have studied: blood group A, blood group B, blood group AB, and blood group O. Here blood group AB is the example of codominance, because in case of blood group AB, you should remember both kind of antigen containing RBCs are present within the same organism. So this is also the case of codominance. One more blood group found in the human being, that is the MN blood group. This MN blood group, MN blood group is also an example of codominance. And one more case I would like to discuss with you, that is the sickle cell anemia. In sickle cell anemia, you remember there are 3 conditions. Normal hemoglobin condition means normal gene, HbA, HbA. This is the normal gene condition, means no disease. One more condition is there, HbS, HbS, mutant gene. Both of the genes are mutant. That is called sickle cell anemia. And one more condition, that is the heterozygous condition, HbA, HbS. This condition is known as a technically sickle cell trait, and this is the example of codominance, because if any patient having one gene HbA means normal gene and one gene HBS means mutant gene, then in such a organism there would be some of the RBCs would be normal biconcave and some of the RBCs would be the sickle shaped. Means two types of RBCs within the same organism is the case of what? Sickle cell trait, means is the case of what? Codominance. So this heterozygous condition for the sickle cell anemia, technically I use the word sickle cell trait, is also an example of codominance. Now on the basis of this codominance and incomplete dominance, I would like to discuss with you two types of questions. Suppose there is a question in a dihybrid cross, one character showing dominance while other character showing codominance, then what would be phenotype of progeny in F2 generation? By using the product law, such kind of a questions you can easily solve within a couple of seconds. Need not to do any kind of a cross. Just remember, in case of dominance, what was the monohybrid phenotypic ratio? 3:1. If character showed dominance, then the phenotypic ratio was the 3:1. 3 dominant out of the 4 and 1 recessive. If character showing either incomplete dominance or codominance, then what was the phenotypic ratio? 1:2:1. Accordingly apply. One character showing dominance means phenotypic ratio 3:1, and second character showing incomplete dominance means ratio second character showing codominance, or if the word codominance replaced with the incomplete dominance also, then also the answer would be same, because phenotypic ratio of both codominance and incomplete dominance was same. So 1:2:1. So the product of these two crosses would be what? 3:6:3:1:2:1. It would be the phenotypic ratio of the F2 progeny rather than 9:3:3:1 that you observed during the Mendelian dihybrid cross. If suppose in this question, so answer for this question, that in a F2 generation of the dihybrid cross, the phenotypic ratio for this condition where one character showing dominance and second showing codominance, answer would be 3:6:3:1:2:1. But suppose in this question I mentioned likewise, in a dihybrid cross, one character showing incomplete dominance while the other character showing codominance, then what would be the phenotypic ratio of the F2 progeny? Then I hope that you all are very comfortable that now this value would also be 1:2:1, and this is also 1:2:1. This is for the incomplete dominance and this is for the codominance, means the ratio would be 1:1, 2:4:2, 1:1, like genotype, means you can easily solve such kind of a questions. Or multiple alleles. If you remember, when I discussed with you terminology, then I taught you that one of the gene that was present since beginning is known as a dominant gene or unmodified gene or wild gene. Remember? Now that gene when go through the process of mutation, either same sense mutation or missense mutation or nonsense mutation, then it gives rise to the modified allele, and that modified allele, if either producing non-functional enzyme or not producing any kind of a protein product, then we recognize that one as a recessive. So I would like to share that one thing, that suppose this is the wild allele. This is the wild allele, and if it go through the mutation it gives rise to the another variant. Then there are two variants of the gene. But is it a guarantee that when wild gene go through the mutation, it will follow only one type of a mutation? It can also mutate with the different kind of a mutation. And suppose if it mutate with the different kind of a mutation, then it can give rise to a different variant also. Now in this condition, within the population, there are three variants of this gene are existing now. That's the multiple allele. Whenever any gene exists in more than two allelic variants, whenever any gene exists in more than two allelic variant, that condition is known as multiple allelism, and such kind of variants are known as a multiple alleles. So you can say that multiple alleles arise by more than one type of mutations into the wild form. Whenever wild gene or unmodified gene mutate by only one type of a mutation, it will give rise to the two allelic variant, that is the normal condition. But if the wild gene mutate by more than one type of a mutations, then the resultant variant would be more than two, and that more than two allelic variant condition is known as a multiple alleles or multiple allelism. So this multiple allelism is also one of the crucial content of the genetics. About it, the features of the multiple alleles and what is their examples we will discuss here. I already told you that multiple alleles are the allelic variant of the same gene. It means they will occupy to the same locus. Because they occupy to the same locus, it means among the multiple alleles crossing over cannot occur. Crossing over always occur in between the non-allelic genes, means the genes which occupy to the different loci. So multiple alleles, among the multiple alleles, there is no provision of the crossing over. One more thing I would like to discuss about the feature, that alleles, one gamete can carry only one allele. An individual can carry maximum two allele. And multiple allele terms we apply for the condition where there more than two variants are present. It means multiple allelism can never be studied within the gamete or within the organism. It always study into the population. Some more features we will discuss in the coming episode. Thank you very much.